Use of bioinformatics. As being an interface between modern biology and informatics, bioinformatics involve discovery, development and implementation of computational algorithms and software tools that help us to get a clear picture of biological processes. Bioinformatics have its own importance in agriculture also. In developing country like India, it plays a big role in agriculture where it can be used to increase the nutritional content, to increase the production, to get a resistance against diseases, etc. In pharmaceutical area, bioinformatics helps to reduce the time and cost involved for different analysis, drug discovery, etc. The use of bioinformatics is threefold. First is to collect and organize the data in a way that allows researchers to access the information and to enter new results as it is produced. Just depositing these data into a database is useless unless it get analyzed. So, the second aim is to develop tools to analyze this data in required way. For example, it is of interest to compare a sequenced protein with the already existing sequence may need a tool or program for the purpose of comparison. Developing this kind of resources can only be done with a deep understanding of computational theory and biology. The third aim is to analyze these data using these tools and interpret the results in a biologically meaningful manner. In the traditional biology, it is impossible to examine the result of an experiment in an extendable way, but using bioinformatics, we can conduct a global analysis of all the available data. The experiment results from biological laboratory will be stored into the bioinformatics databases, and then the analysis of these data can be done by using different computational techniques and tools. The data will be analyzed in terms of sequence, structure, function, evolution, pathways, etc. The result of analysis will be then compared with the biological data and the experiment result. Let us see these aims in detail. The first and core aim is the collection of information or data. So, what does this information stand for? Data can be raw DNA sequences, protein sequences, macromolecular structures, genome sequences, etc. DNA sequences are strings with 4 base letters A, T, G and C, which comprises genes. Next one is protein sequences. These are nothing but a collection of 20 amino acid letters. The most complex form of information is the macromolecular structural data. More than half percentage of this data is the structure of proteins. Genome sequence information is the collection of raw DNA sequences ranging from 1.6 million bases to 3 billion bases. Now, the issue is with the organization of this large amount of data. First of all, most of this data can be grouped together based on the biological similarities. For example, genes can be grouped together according to their function, pathway, etc. So, the major aspects of managing this data is developing methods to assess the similarities between these biomolecules and identifying those that are related. There are different databases available to deposit this data according to the information it carries. Each database contains different types of data. Some examples for the database are protein sequence database, nucleotide sequence database, structural database, etc. So, according to the types of data, these can be stored in different databases. Now, the question is that how can we use or analyze this data in practical life? The information stored in databases is meaningless unless it get analyzed. There is different type of analysis can be done using these data sets. The raw DNA sequence data can be analyzed to find the coding and non-coding regions of the DNA sequence. Protein sequences can be compared using bioinformatics algorithms. This method is using for multiple sequence alignment. Annotation and encoding of sequence is the way to explain a whole genome. As we mentioned before, Our body contains lots of proteins, genes, etc. The relationship between these proteins, biological pathways of different genes, details about the gene products are the core research areas in bioinformatics. One of the major areas of research is at the gene expression level. The gene expression level research is a way to understand the protein and mRNA that are produced by the cell. The main application of this research is in the area of cancer. As we all know that the reason and cure of cancer is still unknown. Bioinformatics algorithms can find the shared sequences between different genes or proteins. The research on gene expression is trying to solve these questions. The use of bioinformatics depends on the area or field we use it. Moreover, bioinformatics has its own importance and use in every field of study. Some uses of bioinformatics is explained here, depends on the branch of study.